This is an Embryology Highlights program. It deals with the early development of the nervous system. The nervous system arises when the notochord induces ectoderm to become neuroectoderm. And these are the stem cells that give rise to the entire nervous system. The neuroectoderm will transform into central nervous system and the peripheral nervous system. And the process by which it does that is called neurulation. Neurulation involves various processes. Here we see that the notochord has induced ectoderm to form a columnar cells, which are the neuroectoderm, and that creates a neural plate. These columnar cells pinch off the surface area and expand at the base, and that creates a neural groove. And the neural groove continues to close as the shoulders come together, and that creates a neural tube. Cells that are at the boundary between ectoderm and neuroectoderm then become the neural crest. Cells that form the neural tube are called neural epithelium. Those cells are going to form the future brain and spinal cord. And as a result of this, we find that the neural tube will become central nervous system, that is the brain and spinal cord, and the cavity will become ventricles of the brain and central canal of the spinal cord. And neural crest cells will become the peripheral nervous system neurons, that is, neurons that have their cell bodies in ganglia of the peripheral nervous system, as well as lemmocytes, which are the glial cells of the peripheral nervous system. This shows the neurulation process that we just looked at in the transverse view, a groove and the shoulders coming together to form a tube and a neural crest separated from overlying ectoderm, all induced by the notochord. This shows that happening from a dorsal view. And as you can see, it does not happen simultaneously along the whole length of the embryo. It begins in the future neck region and zips rostrally and caudally. At the end, there is a opening cranially and caudally, which these openings are referred to as neuropores, and neuropores eventually close. Neural epithelium is the stem cell of the central nervous system. It's going to form central nervous system neurons that have their cell bodies in the central nervous system, glial cells, astrocytes and oligodendrocytes, and ependymal cells. There will be other cells that will come not from neural epithelium. These will be blood vessels and microglial cells. They will invade the central nervous system from mesoderm. Initially, neural epithelial cells contact the inner and outer surfaces of the neural tube, as you can see here. As these stem cells proliferate, they form more stem cells, but they also give rise to neuroblasts, which are going to form neurons, and glioblasts, which are going to form the glial cells. Also, some of the cells will remain lining the neural tube, and those will be ependymal cells. And some of these neuroblasts and glial cells will accumulate and lose their attachments to the surface. So these neuroblasts and glioblasts that collect as a mass of cells within the neural tube wall, they form the mantle layer as a zone of high cell density. So if we look down here, we can see that around the neural cavity there is a collection of cells then, neuroblasts and glioblasts, and this creates the mantle layer. And then the marginal layer outside of that has fewer cells, and the point is that the mantle layer is going to become all of the gray matter of the central nervous system, and the marginal layer will become all of the white matter of the central nervous system. So this is going to be gray matter, and this will be white matter. As the neural tube continues to develop, there is a demarcation between dorsal and ventral, a sulcus limitans, which divides each half of the neural tube into an alar plate, which is dorsal. Alar means wing, and we'll see why that's the case later, and a basal plate, which is more ventrally, and that happens bilaterally, and then the two basal plates are connected by a floor plate, and the two alar plates are connected by a roof plate. And the point is that the basal plate will be the source of efferent neurons that send their axons out into the peripheral nervous system, And the alar plate will be the destination of afferent neurons from the peripheral nervous system, which enter the central nervous system. Down below here, we can see a mantle layer has developed and a little hint of a marginal layer, which is not so obvious. And these are neural crest cells, which are going to form spinal ganglion neurons, unipolar neurons, as well as other cells of the peripheral nervous system. And so their axons are going to come in and enter, and this will be the region of the alar plate that's going to receive input. Meanwhile, the basal plate, which is down here, these neuroblasts are going to give rise to efferent neurons that are going to exit and form the ventral root. Now, as neuroblasts give rise to neurons, those neurons are going to want to participate in circuits. That is, they're going to send processes out to connect with other neurons or with target cells. So the whole idea of being a neuron is to participate in a circuit. And that process is going to be more of a sculpting process. That is, there's going to be an excessive number of neurons that are produced, and these neurons are going to have a profusion of excessive number of neuron processes. And then there's going to be a selective degeneration of neurons and neuronal processes that fail to make functional contacts with other neurons and target cells. So the process is one of elimination of excess material, much as a sculptor would proceed to produce a sculpting project. Neurons need neurotrophic molecules to survive. Neurons that fail to reach appropriate targets will undergo degeneration because they do not receive these neurotrophic molecules. In other words, the targets release neurotrophic molecules, and the neurons depend on those to survive. In the same way, processes of neurons will undergo degeneration if they fail to contact targets and pick up sufficient neurotrophic molecules. So there'll be a selective pruning. Those processes that fail to contact targets will die off. Neurotrophic molecules are necessary then to nurture the neurons so they do not degenerate. Also, neurons release neurotrophic molecules that modify target cells. And so, for example, neurons determine the fiber type of skeletal muscle cells, and neurons modify ordinary epithelial cells to become taste bud cells. So the process works both ways. The way neurons can get their neurotrophic molecules is to participate in circuits, the circuits that are more active, that generate more excitation and more conduction. They'll experience more neurotransmitter. And so degeneration and the survival is driven by neural activity. In other words, it's driven by experience and behavior. So experiences and behavior that are active are going to result in survival of those circuits. The survival of the successful is the theme for neuronal survival in circuits. And those that are less active receive fewer neurotrophic molecules, and those are not going to survive. Below we see the survival process in terms of processes. So this is one neuron in the tadpole rostral colliculus. You can see that we have three processes we're following, and they are color-coded. And we're following these over about a 3-day period. Initially, this neuron gives out this brown process, which is the dominant one, and then a red and a green process are given out, and in fact grow as the brown process begins to fade. So in other words, this brown process was not getting sufficient neurotrophic molecules, and the other processes received enough so that they prospered. And as you follow this along, you can see that the brown process really disappears. The red process doesn't amount to very much. And it really is the green process that survives. So in other words, this green process was making contacts. They were active, and there was conduction and excitation going on. And so that process survived because of the activity and the neurotrophic molecules that were elicited by that activity. We see that it is experiences and behaviors, activity, in other words, that's driving the development of the nervous system. And that remodeling persists really throughout life, mainly at the synapse level. So there's a selective growth and pruning of synapses throughout life, especially in the adult. So the remodeling of neurons and so forth, that takes place in the embryo, of course, and then also neonatally. Later in life, the neuron supply is fixed, and there's a plasticity of synapses. So there's growth and pruning of synapses. Part of this is involved in learning and memory. In general, neurons are incapable of cell division, and so neurons that you're left with following the embryonic and neonatal period of sculpting, these are the ones that you're going to have throughout life. There are some exceptions. In the hippocampus and olfactory bulb, for example, neuroblasts do persist and do give rise to a small number of new neurons postnatally. But in general, neurons that find themselves within circuits that are laid down in the embryo and in the neonate, those neurons cannot be replaced if they pass away. So those circuits are diminished by loss of those neurons. And to finish up, I'd like to go back to this idea of the mantle layer and the marginal layer. And this is the ependyma. So at the neural tube, then, we have this mantle layer and marginal layer. In the spinal cord, you can see that surrounding the central canal here, we have this H-shaped gray matter. And then the marginal layer, the blue here, is the white matter outside of that. So the relationship of canal and ependyma and mantle layer becoming gray matter and marginal layer becoming white matter persists in the spinal cord. So in other words, in the spinal cord, what we're seeing is just a proliferation of the mantle layer and a population of the marginal layer with axons in the white matter. But what about the cerebrum? The cerebrum has white matter, but it's sandwiched in between an outer gray matter, which forms like a bark or a cortex, and inner gray matter adjacent to the lateral ventricle here. And the point is that this develops in the same way. In other words, there's a ependymal layer, a mantle layer, and a marginal layer. In order to get this outer gray matter, it has to come from the mantle layer, and it does so by cells migrating through the white matter out to the periphery. So the same basic arrangement of mantle marginal layer can either form a spinal cord, or it can form two layers of gray matter, an outer cortex via migration and an inner gray matter layer which is adjacent to the canal system, the cavity of the brain in this case. So in case of the cerebrum, this region of the brain needs a lot more cells than it does white matter. On the other hand, if the spinal cord needs a lot more fibers than it needs gray matter. And so the same initial relationship then transforming itself in two different directions according to the needs of the spinal cord and cerebrum. So that concludes our presentation of early embryonic development.